What I saw under a microscope when I had isolated pollen out of Manuka honey. I didn't really understand that inside the pollen there is a process occurring, and the process involves breaking down the phenolics into a polymer, and that polymer is a polymer of glyoxal. And the polymer of glyoxal gets further broken down into CO2 and water as part of the expansion of a pollen tube. So it generates pressure and the liquid water in order to create the tube expansion, which is involved in pollen fertilizing the ovule. So the pollen grains don't start out with this concentric circle of fluorescent light. They start out with a bifringent fluorescence around the edge of the triangular shaped Manuka pollen. So when you isolate the pollen directly from the flower, Manuka flower, you get to see something completely different. So it was only when I found a broken pollen, one that had been damaged, and the damage led to the release of the polymeric glyoxal, which I detected using Maori-Toff mass spec analysis, that I got to see the polymer that was present within the pollen that was responsible for the fluorescence. And then I put two and two together and really started to understand the role that pollen plays in methyl glyoxal formation within the maturation process of Manuka honey. So it wasn't about dihydroxyacetone being present in the nectar that the bees are collecting. It was about the pollen that the bees are collecting and how the pollen changes through a germination process that leads to the formation of this polymer. And the polymer is fluorescent. And then it's released into the honey. And that's where the methyl glyoxal is actually originating from. It's originating from pollen germination processes.